This morning, and thank you, Kirsty, and take it away.
All right. Thanks, everyone who came at 9 a.m. on Friday. So to wrap everything up today, I hope to sort of bring together all of the knowledge that we've learned so far in the last two lectures. And then it's an opportunity to also discuss this in the context of my lab's own work. And this is really thinking about more of this whole organism context and how different species might be able to actuate these elements in different ways for different functions. So I thought I would sort of start with a little homage to physics, I guess. So if you remember these shapes on the left, which correspond to one of the simplest microswimmers, originally hypothetical, that you can imagine that actuates these two hinges with the three links. So of course, these have now been implemented both computationally and also in various guises in artificial devices. And then it's sort of interesting that if you think about this from a purely theoretical physics perspective, construct, or if you're very smart, you can construct this argument, this highly complex argument that essentially boils down to thinking about swimming as a consequence of a sequence of shape changes. And if you deform the body in a sufficiently continuous manner, then you'd expect to be able to sort of move from the first position to the final position via this sort of continuous sequence of deformations. And then there are ways that you could then connect the shape at any given time to the shape at a slightly later time. And this is particularly useful, not just for understanding how microorganisms move, but also for understanding or better designing robotic devices that may be able to actuate or plan their movement at these sorts of friction-dominated, in these friction-dominated regimes. So there's a very interesting implementation, which is actually not directly relevant for fluid dynamics, but nonetheless uses or calls upon a lot of the ideas that we've been discussing so far. And this concerns movement so friction-dominated movement through granular media. And so fairly recently, Goldman et al. Have come up with an argument which suggests that actually swimming or movement through granular media is also associated with this type of resistive force theory argument, where, as a reminder, movement of this sort of segmental bit of rod or segmental body, a slender body through this environment is associated with very different coefficient drag, whether you're moving in the direction in line with the axes of the rod or perpendicular to the axes of the rod. And these graphs were constructed actually empirically from various measurements that they did. I think in this case, towing these rods through granular media. And there's a little bit of a strange way to think about things, but if you can concentrate on this diagram here, what we're comparing is essentially the drag force in the perpendicular direction versus that in the parallel direction. And then you can tow this segment at various angles, denoted by this psi here, through this medium. And so for comparison, in the viscous case, maybe you might focus on this psi equals 0 case, where it's just being towed parallel to the axis of the rod, versus the psi equals pi over 2 case, where it's moving perpendicularly. And there you reproduce the sort of factor of 2 that we saw previously. So in granular media, the ratios are slightly different, but the relative contribution is the same. And so using this sort of knowledge, these authors and over a period of many years have applied this thinking that we think is analogous to swimming through viscous fluids to the idea of locomotion through granular material or sand. And so in this case, this is a really fascinating array of macroscopic animals. These are limbless animals, for example, snakes and all sorts of other sea elegans and similar, they're not too similar, but similar looking creatures. And for the purposes of propulsion, these are limbless, and therefore there is no head associated with these types of movements, which goes back to something that we were discussing back in lecture 1. So if you record high-speed videos, So in order to analyze this, you really need high resolution data. So if you record high-speed videos of these shapes, you can then track these shapes over time at really high resolution and deduce this periodic sequence of shapes that each organism must undergo in order to basically make net progress. And it's useful usually to reduce the dimensions in the system. You don't really want to be dealing with these complex waveforms that you may or may not be able to associate with some sort of analytical expression, such as the one that I showed previously with the sinusoidal sort of wave propagation. But instead, you might try to decompose this complex shape space down to just a small number of modes, so fundamental modes of these shapes. And so this, you may have heard of this approach in the context of looking at C. elegans locomotion. So something like eigenworms, for example, maybe the term that you're familiar with. And these are really just constructed from the principal component analysis. So in this case, there's even a simpler way you could do this, which is just by thinking about the curvature. And so you can really express the curvature, what symbols they use in the paper, but you can express the curvature as a function of arc length and time in the sort of dimensionless, as a dimensionless quantity, and then just expect that there are some sort of, let's call them, this may not be the same notation that they used, but you get the idea that we just decompose this these time series of these curvatures along these two sort of principal components, and these are associated with certain weightings. And so I think what this column is showing here is simply these time series for each of these principal component directions. And so you can actually see that despite the complexity of these shapes, you just end up with fairly sinusoidal looking waveform in each dimension. And then the next step is then to just construct or visualize dynamics in this sort of reduced phase space. So if you then plot these weightings, these linear weightings, then you find that whenever you execute a periodic motion, you're just prescribing a circle in this space. And the circularity is really an indication that we pretty much have sinusoidal curvatures, sinusoidally propagating curvatures. And so, of course, in living systems, you're not really going to have perfect motion, as in these shapes are not going to reproduce themselves exactly. But this is why, yes? Well, I think, yeah, that's a very good point. So typically, a lot of theories become much more accessible if you're just looking at 2D. But whether that's enough is something that you could test. So, for example, I'm not sure if they did this in the particular paper that I cited here, but in the context of C. elegans locomotion, there are various arguments that what you could do is try to add more and more and see where that saturates. So I think in general, for most undulatory dynamics, 2 modes is actually not too bad. But what's interesting, what's interesting is that we're studying this creature here, which is a unicellular organism that remarkably requires more modes, which makes our lives more difficult. But for today, we're just assume there are 2 modes. So that's a point. So when you have this deconstruction, this opens up this, as I mentioned, this really handy tool with which to access control of movement in these types of regimes. And this is exactly the series of papers, starting probably with this one, that makes use of this argument, which I summarize here, which is really inspired by the original paper from Wilczek and Shapiro. And maybe just to simplify things a little bit, let's sort of think about what we actually mean by this shape space. So if you think about the 3-link Purcell swimmer again, you can describe this swimmer or this object completely either by using a sequence of points that correspond to all the positions on the body, which would be quite tedious, or you can just make use of these two angles, alpha 1 and alpha 2, and together with the center of mass coordinates of the objects. And so that sort of simplicity is at the heart of this type of argument. And so what this is the same paper that I cited before. So what the authors did here was they took these sequences of undulatory waveforms corresponding to each of these organisms. And then you can compute using this drag-based argument how much drag force is experienced by each segmental element. And then from that, you can compute the velocity in such a way that the entire system remains force-free. So as long as it's force-free, this satisfies sort of viscosity or friction-dominated locomotion, and this is what they use to construct the directions of these arrows. So you can see that these arrows are not exactly what you'd expect if this were a waveform actuating in a fluid, in a viscous fluid, which is fine because it's not a, it's a solid system in this case. But then the next step is to compute what is known as GG body, the time rate of change of this object here, which essentially allows you to compute how these different parameters change in time. And so the computation of this is a little bit involved, but you can check in the reference in the materials how this was done for these types of experimental data sets. But the point is, once you have knowledge of how this object deforms, so what is its forward speed, lateral speed, so x dot and y dot, as well as the speed of rotation, you can assume using this sort of infinitesimal deformation argument that this quantity can be expressed as a linear combination of the underlying shape space that we constructed previously. And so as a reminder, in this particular case, we're just thinking about movement in the plane. And so this has 3 components. Whereas the shape space really just has 2 components in this case, but you can, of course, change that number. And then the point is, then you have this matrix, which you can start to plot. So this matrix, basically, each row of this matrix is then a vector field. So one of these rows, well, this isn't exactly for this experiment, but you can imagine that for one of the rows, say movement in the lateral direction, you can plot one of these vector fields. And this really quantifies the sensitivity of the locomotion, of the whole organism locomotion, to small changes in this shape space. So at every point in the shape space, you can associate a vector. And then the advantage of that is then you can estimate the sort of displacement per periodic cycle, as you can visualize it as a sort of path and orbit through this space, which is expressed as the sort of integral. And then we're just going to make this very hand-wavy argument that you have the Stokes theorem, but actually you don't because of commutativity issues, which I won't discuss, but you can find some reference to this in that in this reference, and also the references that it cites. So it suffices to point out that this grad cross here, this curl here, is not the classical definition of curl, but it's known as a curvature operator. But the point is then that once you're armed with this sort of very rigorous measure of how much do you travel within any given cycle, which you can think of it roughly as the enclosed area by this orbit, then you can start to set up optimality arguments, which allow you to then say, for any given shape or given gate, where the gate is defined as a particular sequence of shape changes, is there an optimal gate that allows for the best or the furthest displacement per cycle? And so this is quite, again, very useful, particularly in robotics, but you can equally well use this analysis to understand how organisms of all scales locomote through various media. Okay, so at the scale of an individual filament or an individual body part that is capable of instance, this argument or similar arguments gives us a sense of how by changing these gates, you can then manipulate how far you move, but also the direction of movement and all sorts of parameters associated with motion trajectories. And then, of course, the complexity happens when you have multiple of these components, say multiple appendages on the same cell, which may be independently actuated. And then what do you do? And then on top of that, if it's a living organism, it generally has sensors for environmental stimuli. So this could be light or chemical gradients or flows, all sorts of things. So in this, I just found this diagram quite appealing. So in this picture here, you have which is a single-celled alga, which has a single eye spot, which is in red, which it uses to sense light. And so the difference between the self-movement and environmental cues, these objects can then decide, in a way, make an informed choice about whether they should execute a certain type of movement pattern. So in this case, this includes a very sort of intriguing dynamics known as polygonal swimming. So this was, I think, first discovered by Wan III et al. In Nature Physics here. And there they propose a very rigorous mathematical argument for why you get polygonal swimming. But it is really striking when you watch these, when you look at these trajectories, that you do end up with all sorts of polygonal, very polygonal trajectories. Okay, so helical swimming in particular is highly prevalent in nature across all scales, actually, especially in aquatic movement. So one question you might have is, how do different organisms generate helical movement? And often when you track the trajectory, for example, either in 2D or looking at, either in 3D or looking at two-dimensional projections of the three-dimensional trajectories, these look very much. This can be with a single locomotory flagellum, such as in the case of Euglena, or you can have these rather strange organisms. This is a dinoflagellate, which has a single transverse flagellum as well as a longitudinal flagellum. And through this balance of the propulsion generated by these two flagella, you can actually also obtain helical trajectories. And so this is something that we also wondered about in the context of Chlamydomonas again, our favorite organism. So here you have this fairly symmetric geometry consisting of a elliptical body with symmetries, except the position of the eye spot, which will become important. And then it has these two cilia that look the same, and they're the same length. So how do you generate helical propulsion from this configuration? So I don't know if anyone did the exercise, but if you did the exercise, you might appreciate that if you were just swimming with this breaststroke gait, let's say a coordinated breaststroke gait in the plane, then you would move forwards. Okay, in reality, you move forwards and backwards because of the power recovery stroke asymmetry. So what do you need to get a three-dimensional helix? So this was puzzling for many reasons, mostly because of lack of imaging tools back in the day, let's say. So it was possible to obtain these sort of hazy trajectories of these cells, and this is an artist's impression of what they observed under the microscope. You can really get a sense of the regularity in the movement, but there was no obvious explanation for why you would have a helix. And one of the arguments that was put forward in this paper was that maybe the two cilia lose synchronization once in a while, which is actually true. This does happen. But if you imagine losing synchronization on a completely random basis, then there's no way that that is going to translate into a helical movement. So instead, we're looking for a periodic signature. So we can actually find this by modern technology, in a sense. So this time we hold the cell again, stop it from swimming, but observe flagella dynamics from above instead of typically from the side. So if you did that, it turns out that there is this very obvious chirality to the way that the two cilia are oriented. So they even have names. The one that's closest to the single eye spot is known as the cisflagellum or ciscilium, and then the other one is known as the transcilium. And so if you do these experiments, you find that if you localize the positions of the flagella pair over the course of, and sort of bin these positions according to the phase during the cycle, the beat cycle, which we know by the periodic extension and contraction. It's not a real extension and contraction, of course, it's just the observed projection as the cilia execute the power and then the recovery stroke. And so if you put all of this together, you can see that the mean instantaneous beat plane of these two cilia seems to actually rotate. And it rotates in this periodic way that it rotates counterclockwise during the power stroke. So as the cell does its power stroke, it's actually rotating counterclockwise, or the cilia are rotating counterclockwise, which means that if you were not applying this external force, the cell would swim clockwise to compensate. And during the recovery stroke, it's rotating clockwise, so the cell would then spin counterclockwise. And because of the asymmetry, even in this view, then the cell will always spin slightly more clockwise than it does counterclockwise. And so on average, you'd expect it to spin clockwise. And so this kind of explains, we think, why these cells are able to execute helical motion. So you might find this factory because there is no actual model. And so, okay, the experiments would suggest in this sort of fairly limited scenario where we're imaging a held cell from above, that it would also, that were it to do the same thing in a freely swimming context, you would obtain helical trajectories. And so you might gather from yesterday that the fluid flows generated by chlamydomonas cilia is a well-studied phenomenon, and this is true, and particularly in a more theoretical context. And what we did was build on these existing two-dimensional models or configurations of this particular organism and move it to the third dimension. And so essentially what you have here is a configuration of three objects, two small spheres representing the flagella, and then a large sphere representing the body. So the force is really just being, the force propulsion is just being generated by these small flagella spheres, which we prescribe to rotate on these orbits. So remember at the beginning of the lecture, we talked about forgetting, in a sense, the complexity of waveform dynamics and just assuming it can be projected in this sort of fairly low dimensional way. And so you can implement various types of forcing. So in this case, we just assume there's a Stokelet attached or localized to the point of each of these flagella spheres. So this generates, in this case, a tangential force, and this mimics to some extent the swimming dynamics of one of these cells. So you have to satisfy force and torque balance as before. If I hit that too quickly. Oh no, never mind. So you have to satisfy force and torque balance as before, but in this case, we're actually letting this thing rotate out of the plane. So there's a particular angle beta, which I have lost. So I'm going to move this video. Here's the image that was lost. So the idea is that we're rotating, so each of the spheres beat in their own planes. So we're rotating the planes relative to each other, basically. And this angle beta can be then estimated from the experiments. Okay, good, let's go back. Good. So once we implement, again, so once we implement this angle beta, you can see that the whole thing will rotate about its own axis. So this isn't actually a helix. Well, it's a very degenerate helix. And so then the question is, what else do you need to do? So you need to break another axis of symmetry in order to get a helix trajectory. And so this, we think, comes from the imbalance of the two flagellar forces. So if F1 was the same as F2, the thing would just rotate axially. But if F1 was not the same as F2, you start to get all sorts of interesting helical or superhelical patterns. And so there is evidence for this from a physiological perspective. So for example, if you imagine the reason for not just generating helix, but also why do you need to have an asymmetry in the two cilia? This comes down to the fact that if you place many of these cells, this is a high density suspension of these algae in a dish, and you shine light from the side, and then these cells will actually all migrate towards that light source. And then this is obviously indicated by the concentration. And at a single cell level, this happens because of the asymmetry in the two cilia. So there's a fairly complex phototransduction pathway whereby light is detected by this eye spot, and the signal, a bit mysterious at this point, is transduced to these two cilia in a way that is imbalanced, and this allows for an asymmetric turning event. So, and it turns out that in order for this to happen, there is an intrinsic asymmetry between the two sides. So even in the absence of any signal, if you're just imaging the cells in a uniform background, you would still have the same underlying asymmetry. And so this can be implemented in different ways, but in our case, it's just through the magnitude of the force, which results in a different rotation frequency of one of the cilia. Yes?
But then this means also they cannot swim in a straight line, right? They have to.
Yeah, so they never really swim in a true straight line, which is kind of interesting. So if you, so in this first example here, that's a very unrealistic example. Most of the trajectories that you measure in nature look something like this. So they're very tight helices, but nonetheless, there is a small asymmetry. Yeah. That they don't synchronize, but yep, definitely. Yep. Yes, well, this is basically this was my PhD project. But let's not discuss that again. But I think the point is, I'm just going to summarize some years of work, not just from our side, but also from others in the field. So you can, and actually the authors I cited here, they implemented this exact scenario, but actually they did have a sectional dimensional component, but it was not, let's say it was a simpler version of what we now know to be the case in terms of how the flagella are actually actuated. But in this context, they did implement what would happen if you have asynchronies in the beat pattern. So this is important, but I think we wanted to study what would happen in the absence of these asynchronies, which we know through other studies are always happen in a very stochastic way. So we were kind of focusing on what do you need to generate periodicity. But it's a good point. Another question? Yes. Yeah.
To make it to whatever the motor system.
Yeah.
There has to be something true about the scales for the signal.
Yeah. So there are some studies over the course of 30 years or so that basically show that in the case of Chlamydomonas and in many other algal flagellates, they may have different configurations, but they share this ability to respond to light and then swim to the light. And so in all of these cases, there's a very sort of, the frequency of body rotation, which arises, we think, from how non-planar the beat pattern is fine-tuned, presumably by evolution, to be optimal for sensing. Given the parameters that you mentioned in terms of time scale for signal transduction, etc. So it's and of course it's important also to keep that rotation timescale far away from the intrinsic timescale of flagella oscillations. And so in this case, the oscillations occur at around 50 to 100 beats per second, whereas the body rotation is only around 2 hertz or less. So I think this is, it's quite a clever way if you're designing a system that is to be robust to photo sensing, to design it in this way, I think. Yes. Yes, and various people have studied this, but it's actually quite difficult to do the experiment well, because often the people that are interested in these types of questions are biologists, and what they tend to look at are experiments like I showed here, where you just have lots of cells in a dish, and then you get a sort of population level quantifier for does this thing do phototaxis or not? How well does it do phototaxis? But if you really wanted to address that properly, you need to do this three-dimensional analysis or imaging. But I think this is very difficult in general. But yes, the answer is yes, it does make a difference. Okay, so with this of, configuration, you can then imagine various functional forms for how the signal, let's say, the signal from a light source is detected by the eye spot, and how it then in turn changes the relative balance of the two cilia. So in this case, based on our biological knowledge, we assume that this quantifier of asymmetry depends directly on the intensity of the light source. And so at some point, you would have this separation or this divergence in the response. And so the more light, approximately speaking, the more light you have, then the greater this asymmetry between the two cilia. And this is something that has been observed biologically. And so we think that then this mechanism, coupled to the three-dimensional rotation, then underlies the cell's ability to do phototaxis in 3D. And this is really smart in a way. You don't have many components to work with. As in, there is no, and probably this is something that this type of thinking is something that it will preempt Manu's presentation probably later in the week. Is this idea that you just harness your body, basically minimal components associated with the configurations of the sensors on the cell and the shapes of the organelles and the filaments in a way that allows you to just have this robust response without needing control. Okay, so, and then yesterday we started talking about synchronization and coordination of cilia. And we said that if you have two cilia, there is some hydrodynamic coupling that always exists because they're in the fluid that under some circumstances will be sufficient to coordinate them. And then I had many questions at the end about, okay, but what happens if you have many cilia? Okay, so this is what my attempt to answer. I'm not going to give you it's a bit controversial, but I'll attempt to answer this through a series of arguments. So on the left, you have paramecium. This is a very high-speed video, so it's very, very slow. Meter a second or so it's actually quite fast in reality. But you can see the deformations of the tens of, probably hundreds of thousands of cilia on the surface. And then animals do this, also mitochondrial waves of the limbs. But then if you try to manufacture, as I hope I cited this, as these authors did, some sort of ciliated carpet, this is actually quite challenging. Of course, the authors will not say it's challenging, but relative to how it's done in the cell, I mean, each of these elements has to be actuated by magnetic fields. And so if you imagine then there is some sort of change in the environment, okay, but then the person doing the experiment will have to change the pattern of actuation of these magnetic particles to make the thing swim or move according to a different gate. But the organism doesn't have the luxury of that. And so how do they do this? Is there some sort of spontaneous coordination that arises because of just the fluid? So here are some organisms that we study in the lab. And so I just put this up here because I think it spans some of the different regimes that you might encounter when asking this particular question. So when you have just two very small, very long and slender cilia in isolation, they will move together, as we saw yesterday. If you have an array of these, they will often exhibit metochondrial waves. And then if you have even more of these, so this is an animal larva, the wave is even more, let's say, spectacular. But it has many interesting features that I don't really have time to discuss today. But if you're interested in this general topic. So the reason why we study this really manyfold, because we find these types of systems in everywhere, not just in small organisms that we study for the sake of biophysics, but also because of that they exist in various vertebrates, such as ourselves. And so you can find all sorts of configurations of these configurations of these arrays of cilia that are relevant, whether for swimming, mucociliary clearance, or for feeding potentially. And they almost always share this feature that there are coordinated waves of activity whenever you have an array of them. So in this sort of picture, imagine that each element is doing this power recovery stroke, this asymmetric pattern, and then you place them on this grid. So then the metachronal wave is then defined by various parameters associated with this movement. So whether you have a phase difference between neighboring elements, and in which direction is that phase difference. And it turns out that in nature you find all sorts of organisms that exhibit different kinds of metachronal coupling of the cilia. And so I wanted to show this slide just to sort of put the words in your head. And these types of synchronization or types of metachrony have been defined. I think this is maybe the very first paper to define these terms. And so the basic idea is that you can have one of 4 different types. So if you imagine the direction of the power stroke, which is also known as the effective stroke, and the direction of the wave, then either these things are in line with each other, in which case you would have either symplectic metachrony or antiplectic metachrony, depending on whether they are parallel or anti-parallel vectors. And then, of course, you can have it to the side or at some angle relative to the stroke itself. And so this collectively, this class is known as a diaplectic metachronal wave, which can be either leioplectic or dexioplectic. So there are many types of metachronal waves, and you find those in other animals as well. This is just a nice animation. And it's quite fascinating to study why there is so much diversity. Yes, I have a slide about this. So you might ask, okay, but what is the purpose of these waves? So many, some authors have tried to tackle this problem using computational approaches. So what you can do is, in these computational frameworks, assign or input the waveform that corresponds to a single element, one of these things, which you can take from the data, let's say. And then you can arrange these according to different patterns. You can put them on the surface and ask how much flow is generated. So imagine the cilia in your lungs that are responsible for mucociliary clearance, then the more flow, the better. And then you can otherwise put them on a sphere and ask how fast does it swim, which may be an important question if you're a microorganism. And you can ask, well, what is causing the synchronization in the first place? And so there's a wealth of literature on the subject, if you're interested, that I don't have time to summarize in 3 minutes. But you can probably appreciate that there is no, as much as physicists would like, there is no single answer to this problem. So there are certain types of metachrony that enhance some functions, not others, and vice versa. And so one thing that, one paper that I did want to highlight is this one here, where what they did was arrange an array, in this case, a linear array of these cilia-like oscillators, just like in the Chlamydomonas model that I discussed before. But in this case, these cilia are, again, they're moving along circular orbits, but they're coupled through this hydrodynamic interactions with the neighbors. So in this case, if you start from so on the y-axis, you have the cilia or the number of the oscillator, and then the color represents its phase. So you start from this fully randomized configuration where all the oscillators have a different phase, and then you just set them going with hydrodynamic coupling between them. And then over time, conveniently no axes here, over time, you start to see the emergence of these propagating waves. And so it's really possible for these types of systems to exhibit spontaneous metachrony. But is it really enough in general? And I would argue no, but I think I'll stop that controversy here. So with that sort of idea in mind, how do you actually organize these appendages if you can't rely on the flow? So what other ways are there? And so in general, this brings us to the study of animal gaits and centipede of waves. Limbed animals also have gaits, and generally these are thought to arise or are related to the patterning of these so-called central pattern generators inside the vertebrates, especially the vertebrates. And is there something equivalent at scale? Could there be something equivalent at the scale? So what you can do is then study a carefully chosen array of organisms that have increasing number of appendages. And this one is Chlamydomonas. You have the idea that they just swim with this coordinated breaststroke. But as you start to increase the number of appendages, maybe the task becomes more difficult. How do you, when you have so many arms and legs, how do you coordinate them if you don't have a central nervous system? And when you have too many of these, then inevitably these appendages start to couple through the fluid. And so this point is really a question of competition between what's happening inside the cell, what the cell actually wants to do, and what the fluid wants to do. So how do you maintain that balance, or how does a living specimen even change its behavior depending on the environmental circumstance? So one sort of fairly, let's say, powerful experiment that you could do is the following, where you take a cell that has ordinarily 4 of these appendages that are actuated in some sort of gate. And then you fix it, stop the body from moving. You first check that the appendages move in a certain sequence, and then you stop one of the cilia from moving. And so you basically remove all of these points out of the equation and basically ask what's the order of the remaining appendages. So the point is that there is no change to the order of the gate. And so there is really something inside the cell, nothing to do with the fluid around it, that is setting the particular gate coordination sequence. And so you can do other types of experiments on a similar vein, where this is, I think, the same. Now it's a different species, but you can see that by manipulating the environment, you can actually make them turn on and off their motility dynamics. And this slide here is just a summary of one of these experiments where if you watch, say, that cell from the side, you can only keep track of 3 out of the 4 appendages because the other one is on the other side. And then you can, again, keep track of the phase, just like the theoretical study did with the phase oscillators. So you keep track of phase during a typical cycle, and then you can see that there are oscillations which stop and start and stop and start. And if you zoom in, this is getting very obscure. If you zoom in, you find that actually these individual oscillators will start and stop independently. And so what this suggests is that actually these cells, they know which oscillator is which. So there's a sense of identity, which is quite surprising at this scale. Okay, so given that appendages generate movement, and by changing these gates, you can change the movement. So naturally, that leads to how do you change your trajectories on a global level? So this is the classic reference from Howard Berg, where, as most of us will know, how bacteria and E. coli in particular will do this run and tumble locomotion, where it will alternate between periods where it just swims straight versus periods where it changes direction. So in general, the argument or the reason for why E. coli and many other species of bacteria have to do this in the first place is to make use of this algorithm to do things like chemotaxis. And so by modulating the probability of these tumble events, it's then thought that the cell can then move towards, effectively move towards the gradient. But actually, if you think about it, this is not a real taxis in the sense of a gradient following strategy. But I think it's too late to change the name now. So there is a fundamental distinction between chemotaxis, as we tend to perceive it, and phototaxis that I mentioned before, which is a much more deterministic strategy. So during phototaxis, a chlamydomonas cell, let's say, will actually orient itself towards the gradient, whereas an E. coli generally doesn't do that. But the point is the same. You can achieve many different patterns just by changing. Oh, sorry, question. Yeah, okay, so this is, yeah, so I think this comes back to a question of terminology. And actually, I was in Germany a few months ago at a chemotaxis conference where we spent an hour collectively discussing the term. I mean, we didn't have we actually didn't come up with a definition, but I think generally it's just good to be aware of that distinction. Maybe I think just from the terminology, because taxis, yeah. I mean, I think just generally in my head anyway, and according to half the people at the conference, you need a gradient following strategy. So it's not so much a stochastic strategy where you eventually end up in the right place, because so there's another term in the literature which is chemokinesis, which involves for example, just changing speed in response to a chemical gradient. And so if you imagine, the swimming speed is some scalar times its direction, in that case, it's only the scalar part that responds to the chemical gradient and not the direction. But I think in a real taxis, but actually we have experts on this here, so yes, yes. I think that is another useful distinction. I mean, I think this also comes down to why you hardly ever see truly phototactic organisms at very, very small scales. But I think, yeah, there's definitely that distinction as well to be made. But yeah, we can discuss more as well. Okay, I hope I haven't said anything too controversial, and I'll regret it later. So the point is, then you can have many different species that are then individually capable of changing the gates in different ways. So how does E. coli do this? So this is the most well-known example. And you have these grainy videos from Howard Berg. This is a beautiful drawing that interprets all of this information. Basically, what happens is that, as you might remember, these things rotate. By rotating a helix, you swim depending on the handedness of the helix. In this case, if you have a single, say, left-handed helix rotated counterclockwise from the back, then it swims forwards. And then if you have several of these merge points on the cell, and there's an argument about whether, I mean, it's probably highly likely that because of hydrodynamic interactions between these individual helices, they tend to bundle. And if they bundle, they basically become this superstructure that then rotates in the same sense, and then that helps the thing propel itself forwards in a fairly straight line until, because of stochastic reasons, these things start to spin, the motors spin in the opposite direction. And then this causes the bundle to split apart. And then you have these transients, which causes a change in the direction, and then the cell swims again. So the advantage of this, going back to the very first slide in the first lecture, we know that this basically results in a diffusion process. And this is associated with an effective diffusivity, which is given by the square of the speed characteristic run speed over the characteristic time. And then generally, this is much bigger than you would just get from thermal diffusion. And so obviously, it's good to swim for these bacteria. If the objective is to somehow navigate a greater. So the second example is a little bit less well known and certainly not understood very well from a mechanical perspective. And this is something called Campylobacter, this horrible thing that lives inside you. And I'll show the movie. It's a really nice movie, actually. They can label the flagella and the body independently. Sorry for anyone who's colorblind. And the weird thing is that each of these cells, still rod-shaped, has two of these flagella, one on each end. And then the two flagella have the same handedness. But then on top of that, the body itself is also a helix, which with the opposite handedness. It's really hard to wrap your head around what's happening. But if you read the paper, they say basically that in order to swim forwards, one of these flagella is actually doing the propulsion, while the other one is wrapped around the cell itself, the cell body itself. And then whenever it needs to change direction, the other flagellum that's not currently used for propulsion starts to reorient. And then you can see that this somehow probably dynamic instability then drives this new configuration. So there's a lot of interesting arguments to be made about why is this actually useful, and could it not be simpler just to design I mean, why do it in this complicated fashion? Why not just have a single cell with a single helix? Is that not equally efficient for swimming? Yeah, I don't have the answer to that. And then the third example is from the world of eukaryotes. So this is another organism that we use a lot in the lab. So this is Pyramimonus, which is another green alga, which has 8 of these cilia. Sometimes it's stationary, looks like this from the top. sometimes it runs forwards, where the run is an analogy of run in E. coli. But in this case, all the cilia are coordinated in a very specific way. And then once in a while, it goes to the front of the cell, causing the cell to move backwards. So you have all these transitions that become possible. And there was some question yesterday about whether there are organisms that can switch between puller and pusher modes. So actually, this is one of them. So just to look at it again. So here's the sort of classic puller configuration, and then it becomes a pusher transiently, and then it goes back into a puller again. But these are very stereotyped events, and also there are stereotyped transitions as well that are possible, that characterize each of the species that we study. But the overall point to all three of these examples is that organisms generally have a way to change direction. And this is probably critical for dispersal. And then the next thing, going deeper into this question of, okay, so there is some vague idea that something inside the cell causing these appendages. Could there be some signaling that further regulates these processes? And so for this, we turn to a different organism together, but these are still low Reynolds number locomotion, and this is something called euplotes. It's a single cell ciliate. This is not a cockroach ciliate side. You see that it rests sometimes on debris with its many appendages. So from the side or from the bottom, it has precisely 15 of these appendages called cirri, and more cilia that are used for feeding. So it has many cilia, and it has many different types of cilia as well. So this is the dorsal surface of the organism. You can see these other types of cilia protruding from the surface. But the point is, underneath, you have these huge bundles of cilia which make up the entire, each of these entire cirri. So each of the legs that it uses for walking is about 50 individual cilia. Okay, so how does this thing, so that's the context, how does this thing move and coordinate its behavior? So under the microscope, it looks like this. Nothing very spectacular. But then if you zoom in with a high-speed camera, this is what you might find. So this is from below. You can see these things walk somehow in a straight line. And then once in a while, it will turn as well. So I'll maybe skip ahead to this bit of the video where it starts to turn. Wow, it's a very high-speed video, as I said. Oh God, it's taking a long time. Okay, there we go. So you can see it turn. And these turning events are actually also very stereotyped. So then the question is, what's causing this? So what we can do is then, this is a hypothesis based on decades of literature, that maybe the cell's membrane potential is responsible for coordinating these appendages, or has some role in changing the dynamics of these appendages. And so what you can do is then build a electrophysiology rig, and then you record on a single cell basis the membrane potential electrical activity. So basically, you hold the cell again to stop it from moving, and then you basically access the membrane potential with another electrode. So this electrode goes inside the cell. Yes, it's probably suffering. And then you can measure the changes in the dynamics of the individual cirri over time, but then at the same time, you can record its membrane potential activity. And so you have to sort of familiarize yourself with the number of appendages that it has, and also the identity of these appendages. So maybe I'll just go back to the drawing again. So it has every cell looks like this. It's already quite remarkable. So it has front serae, about 10, well, precisely 10 of these front serae, which we think are used for forward walking. And then there are 5 of these back serae, which are only really active during turning events. So if we go back to this picture again, you can track these front serae, which the activity of which becomes upregulated whenever a membrane depolarization. So these are not true action potentials like you might find in neurons, but nonetheless, they are associated with specific changes in the value of the membrane potential. And then you can also look at what happens with the back serae, and they seem to be doing something that is opposite to what the front serae are doing. So that's kind of strange. So then we can track, this might be easier to explain with a video. So you can track the dynamics of these, keep track of these individual serae. So this is the front of the cell. At the same time as the membrane potential, you focus on this line here, which is time. You can see that in the baseline, this is the baseline scenario, which is associated with a particular sequence. And then when you have a membrane depolarization, these things start to change. So you have to convince yourself that there is a change. And then if you keep watching this, the change is very stereotyped, basically. And you can also capture the cell from a different angle. So this is now holding it from the back. Well, dorsal ventral is a bit ambiguous in this context. And then you can see the back serae and the front serae. Sorry, the front serae and the back serae. And again, there are stereotype changes in the activity of these appendages. To make this more rigorous, you can track them or attempt to track them. And this is actually surprisingly difficult. So I don't know if anyone actually studies animal behavior here, probably not. Oh, question, sorry. Okay. Yes. Yeah, but I mean, well, milliseconds for each individual event, I guess. Yeah. Yeah, that is, yes. And also it should be the same everywhere for another reason, because it's the same, it's one cell. Right, exactly. Yeah. I mean this is one of the mysteries that that we don't manage to answer in this paper, because I mean, like all good mysteries. That we have some ideas for why you might have this asymmetry or difference, and I think it's sort of similar to the case of the chlamydomonas cell that I mentioned earlier. There you have these appendages that are apparently the same, but there is some underlying signaling that causes an asymmetry. So we think maybe that there is an asymmetric distribution of ion channels inside the back and versus the front end. I mean, does anyone if anyone knows a way to measure this, I'd be very grateful. But otherwise, remains a hypothesis. So as I was saying, we then try to track these appendages. So if you work on animal behavior or something like this, then you might you might have come across all sorts of interesting AI-based tools that allow you to track appendages, etc. But they work surprisingly well on these affinities because of the fact that they are many different appendages of interest. They all seem to overlap and they deform, and there's no fixed number. But nonetheless, in a limited situation, you can actually track them by assuming there are, in this case, we assume that there are a total of 4 different animals associated with each individual cirrus. And then what's interesting here is the signatures of non-equilibrium activity here, where if you focus on the baseline state where the potential is kind of at this baseline level, and then you kind of build up this picture of how often do you traverse, or how often do you land in a particular location in this phase space consisting of, in this case, probably 2 angles or 2 positions. And then you find this very pronounced circulation. And of course, you could assume or try to some parameter like entropy production, etc. But the point is, this picture becomes very different from what happens when you have a depolarization event. And so the point is that cell has these 2 states regulated by membrane potential that correspond to 2 different waveform signatures. And then, just to make it clear, you can track this in time as a time series. And if you zoom in on one of these depolarization events, you can see that an individual cirrus will transition from this sort of mode to this more attenuated mode and back to the original shape again. And so if you bring all of this together on a whole organism context as promised, so then you have this picture where basically you have a cell that has some number of appendages that it can regulate. And during the baseline state, it's only the power stroke or the front appendages that are relevant for motion. And it has this particular waveform, which has an asymmetric pattern, just like most cilia do. And this is associated with forward propulsion of the cell. But during a depolarization event, what happens is that suddenly the back cirri start to move in this way that causes, sorry, this is the back end. So the back cirri start to move in a way that causes the cell to move backwards. So this is a short-lived transition before the front cirri take over with a slightly different pattern, which causes the whole cell to rotate. And so basically, this switch, this electrical potential switch, allows the cell to transition between so-called forward walking phases and these sidestepping reactions that are highly stereotyped. And so you end up with what is effectively run and tumble, but a more deterministic version. And this is also a eukaryotic organism. And so if you really wanted to, you can convince yourself of what I just told you. This is essentially subtracting the centroid of the center position of the cell, and then just convincing yourself, okay, which is the front end, which is the back. Well, this is the front end, this is the back end. And then you can see that particular sequence that I described in action, effectively in the frame of the organism. Okay, so I have a few minutes left. So I wanted to also then put this into context. So you have all of these different organisms that each has a stereotyped motility pattern, and most of them, you can call it all sorts of different names, but most of them have this shared feature where they do some sort of forward movement, which is fairly steady and longer lived, and some sort of transient, which may be a tumble or a reversal or a shock or something like this. And the consequence of that is generally to create a biased random walk on average. But then if you are a slightly larger organism, does the same strategy actually mean something? Does it actually have a slightly different consequence for the overall ability of the organism to perform sensory functions? So for this, we like to show this slide here, which summarizes the diversity of what is actually possible. So these contain pretty much all of these organisms at the relevant scale that we're interested in, where on the x-axis you have the size of the organism, and then on the y-axis you have its swimming speed or locomotion speed in general. So you have this, I included this patch of cells or patch of organisms, which contains everything to do with eukaryotic chemotaxis in the classical sense of large cells that migrate, for example, inside tissues. So these typically have large size and move fairly slowly relative to swimming. So up here, we have in the cyan cluster, very small cells such as E. coli and archaea, which are very small but fast relative to its size. And then here we have this distinction between ciliates and flagellates, which are all eukaryotes that have an order of magnitude increase in size, slightly higher speed, but they also cluster. And then the fundamental distinction, well, before I go on to that, there are some lines here that I drew. So here is the Reynolds number equals 1 line, which confirms that all of these points lie below the line, so they're in the correct regime. Here is the Peclet number equals 1 line, which is just the same, similar to the Reynolds number in dimensions, is UL over the diffusion instead of UL over the viscosity. And so this suggests that everything above this line can improve its ability to acquire nutrients by stirring the environment. And this is pretty much everything that has cilia can be located in that region. And then there's another line. So conveniently, these are double log axes, so everything becomes a straight line, which represents the line that separates spatial versus temporal sensing. And so this kind of gets to one of the questions that was asked earlier. In which regimes is one strategy better potentially than another? So why did I draw this line here? I think we do have time to go through this argument, but I also wanted to just highlight another feature of size, which is the susceptibility of these cells to rotational diffusion. So if you're a bacterium, you're very easily pushed off course just by thermal fluctuations. And so for bacterium, or anything that is of the size of a bacterium, it's very difficult to maintain your trajectory. So you can't swim in a straight line for more than a very, very short period of time. If you're just 10 times bigger than this, so if you're a cell of the size of a chlamydomonas or similar, then suddenly the rotational diffusion is reduced by 1,000-fold. And so suddenly you can, assuming you have the right apparatus to sustain this movement, you're able to then swim in a straight line. So on top of that, if you then build in other capabilities such as sensors or actuators, then this allows you to maybe construct a better algorithm for navigation in general. And so I just wanted to sort of maybe highlight some of these in these cartoons, the diversity of different locomotion strategies that actually exist in nature. So all of this will be bound by the same physics that we just talked about over the course of this week. And all of these are actually in a slightly different physical regime, is my final point. And this slightly different physical regime that becomes accessible by virtue of its slightly larger size, and also these appendages called cilia that, as you might remember, have many more degrees of freedom for deformation than just rotating a rigid helix on a rotor, then allow this collection of properties then will allow these organisms to have more intelligent functions than maybe their prokaryotic counterparts. So if we wanted to really think about all of this from the perspective of sensing and understand why is it that I drew this line here, where the claim is that everything above this line should be better off sensing using temporal strategies, whereas everything below this line, such as eukaryotic chemotaxis, should be better off sensing using spatial strategies. That one argument that you could put forward is really just a matter of counting molecules. And this originates from classic work from Berg and Purcell et al., where the basic idea is that in the context of chemosensing for the moment, the basic idea is that you're just trying to sum up the number of molecules in the environment, which is directly proportional to the concentration gradient of this chemical that you're trying to sense. So for this reason, you can construct the scaling argument. So the number of molecules, let's say, inside the volume of space occupied by the cell should scale as the concentration times the volume. And then we know, or according to the central limit theorem, you have giant number fluctuations where the noise in that number will scale as the square root. And so this leads to a signal to noise ratio that takes this particular form. And then the argument from Berg and et al. Is that one of these organisms can then improve its ability to count by just making multiple measurements, multiple independent measurements over time. And the number of measurements that it's able to make, let's say in a time t, is just how many, you just count up how many sort of diffusion timescales exists, where the diffusion timescale here is how long does it take for a particle to diffuse outside of this small volume. So this is then given by the square of the size divided by a characteristic diffusion. And so if you then take the whole, take that as t over this quantity, this gives you how many counts are you likely to make. And so the consequence of that is then you reduce the error in the counting significantly. And so I don't really have time to go into too much detail, but you can try this at home. The basic comparison is then one between a temporal evaluation of the chemical environment versus a spatial evaluation, where the temporal comparison is where you're just imagining an organism in motion, and then you have receptors all over it. And if you fix the location of the receptor on the organism, then what is the count at the same position over different times, over successive times as the organism moves, versus in this scenario where you fix time and then just measure, for example, the difference between concentrations in the front of the organism versus in the back of the organism. And then what drops out, if you calculate the signal to noise ratio in each of these contexts, is a ratio of signal to noise ratios, which scales as this thing here, which is exactly that I plotted, which separates these two regimes. So the point is, if you are a slightly larger organism, then spatial sensing is better. Okay, so finally, the last point, this is really getting very philosophical and controversial, is this, I pose it to you to think about, do we, maybe given all of these complexities associated with unicellular organisms, both bacteria and also eukaryotes, could it be that in the course of evolution, in order to learn, or in order to understand how to control these different appendages for motility, that these organisms may have acquired some semblance of biological cognition? So this recognition of what is actually the self versus the environment. Anyway, so I think I'll just summarize what we learned so far. So locomotion is constrained by physics, surprise. But it's interesting that there is evidence of convergent evolution, where you have all sorts of appendages that basically have very similar physical characteristics. Obviously, the mechanism by which these appendages are actuated will differ significantly across phyla. And then the point is, when you start to ask the question of how different species control self-movement, you have to think about the biology as well as the physics. And so with that, I'll just end. Thank my current lab, various collaborators, some former collaborators that are no longer on the slide. We're also recruiting if anyone's interested. And yeah, thanks very much. Last question. Yeah. Yeah. Uh, 1/8. Oh, you mean the video where they this one? Sorry, the question again. So you think it's I think it's probably less than that. It's probably less than 1/18. Yeah. It's it's an interesting point, but probably, I mean, the thing to note is that most of these species have a set number of flagella, which in bacteria species, it can vary within a spectrum. But these things always have two, this particular species. But you can maybe ask the same question about maybe E. coli. Does it help if you have more flagella? Does it help turning? Does it help the cell to turn? And I don't work on bacteria. Maybe there are people that know the answer. But I think people have studied at least the effect of the number of flagella on the speed of swimming. And I don't remember what they found. But it's an experiment that is feasible. Yeah. 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 It has feet. Yeah, good point. Or, I mean, this is without really thinking about it, but maybe that there's just friction. So, when it starts to move beyond a certain amount, then it's able to depart from the surface. There is definitely still adhesion as well, but something that we haven't measured. Yeah, it's sort of mischievous of me to sort of just put this in this lecture, because it's not really swimming exclusively. But then again, I also included these some things that move in sand. So I think the point is that it's still the model, from the model that we built, it's still able to exploit this drag anisotropy because it's fundamentally still friction dominated. And so, you know, there may be subtleties associated with the choice of place for settlement and things like that, but so far we haven't really looked at that in detail. Yep.
The animal that had like many different legs, like front and back ones, and it
Yeah.
was controlling for acting. Did it control like it controlled like when it would start turning, but did it also control like how much it turned or like how long it was turning for?
Yes, right. So in this set of experiments, we only looked at what happens under no environmental perturbations. So it's just a, I mean, of course, there's going to be environmental noise, but if you measure, say, the statistics of turning, it's actually very stereotyped. So you get the same, pretty much the same turning angle with some Gaussian distribution. But I think, you know, we haven't measured this systematically. If you then change the environment, that distribution does change. So it's possible that the organism, I mean, it's very likely that the organism has the ability to regulate how long it does this depolarization for.
If you make the environment.
Viscosity, but also chemical environment as well. Any other questions? All right, thank you.